Welcome to the deep dive. We cut through the noise, getting you those crucial insights from, well, groundbreaking research.
Straight to your ears.
Exactly. And today, we're kind of getting our feet wet, you could say. We're diving into a really foundational 2016 paper.
Ah, yes, the Deiner paper. Environmental DNA reveals that rivers are conveyor belts of biodiversity information. Christy Deiner and colleagues, a big one.
It really is. So our mission today for you listening is to unpack why this study was such a game changer for biodiversity assessment.
Yeah, it's really the study that took environmental DNA, or eDNA, from this interesting theoretical idea to a practical tool you could actually use at scale.
Right. So maybe let's set the scene a bit. Before this paper, what was the biggest headache for ecologists trying to figure out what lived in, say, a whole river catchment?
Oh, it was fundamentally about scale
Yeah.
and logistics. I mean, you just couldn't sample enough. Think about the standard method, right?
Yeah.
The kick net.
Okay.
Stand in the stream, kick up the bottom, catch what flows into your net. It works. You get a good snapshot.
But just for that one little spot where you're standing.
Exactly, just that tiny localized patch. And that leads to what we call spatial autocorrelation biases. Basically, your samples are all clumped together. They're related just because they're close.
Uh-huh.
Which means you consistently and often significantly underestimate the total biodiversity across the whole system. It's chronic undersampling.
So if you wanted to know everything living in the entire Mississippi River Basin, you'd need, well, an impossible number of kick nets.
Precisely. You needed some way to get an integrated sample, something that reflected the whole system. But the old morphology-based methods just couldn't deliver that.
Which brings us right to the core idea of this study, this conveyor belt concept. They hypothesized the river itself could do the collecting for us.
That's the elegance of it. The idea is simple, really. Every organism, whether it's actually in the water like a fish or, a deer drinking on the bank, is shedding DNA.
Right, through waste, skin cells, bits of tissue.
Yeah, all that stuff. That's eDNA. And the hypothesis was that the flowing water doesn't just wash it away and dilute it into nothing. It actually aggregates it, collects it, and carries it downstream.
Like a conveyor belt.
Exactly. So a single scoop of water downstream potentially holds a mixed signal, a composite picture of all the organisms way upstream, even across different environments, the land-water interface.
Okay, that integrated signal, that sounds like exactly what conservation folks need. Why is getting that bigger picture, that broad richness estimate, so important compared to just knowing what's in one spot?
Well, conservation is often about making tough choices, right? Where do you focus your limited resources? You need to identify the biodiversity hotspots, the places with the most variety of life.
Makes sense.
But if your traditional methods are always lowballing the richness because they're too localized, you might completely miss a really critical area. You just can't get reliable large area estimates needed for, say, regional planning with kick nets alone.
And it's not just about better science. There's a huge cost factor, too, isn't there? Shifting to just grabbing a water sample, that sounds way cheaper.
Oh, massively cheaper. The scale of current monitoring is, well, it's pretty staggering. The paper points this out just looking at the U.S., England, and Switzerland combined. They're spending somewhere between 117 and 207 million U.S. dollars every year.
Wait, hang on. Over $100 million annually. Just for aquatic biomonitoring using these traditional methods we know are probably undersampling.
That's the figure they cite. So the potential cost saving is enormous. And it's not just cash. The researchers highlight 3 big practical wins for eDNA.
Right.
1, it's non-lethal for almost everything. 2, you barely disturb the habitat. And 3, because it's one single protocol collect water, it simplifies everything. It's more cost-effective, less need for loads of different taxonomic experts for every sample type.
Right. You don't need a fish team, an insect team, a plant team.
Potentially, yeah. It
Right.
streamlines it.
Okay, so the motivation scientifically, economically, ecologically, it's super clear. Let's get into the nuts and bolts. How did Deiner and her team actually test this conveyor belt idea?
They set up their study in Switzerland in the Glatt River catchment. It's a subcatchment of the Rhine. And they picked 8 specific sites, sort of nodes along the river network, started below a lake, then followed the main river and some tributaries.
And at each site, they grabbed water samples. About what, just under a liter?
Yeah, 840 to 900 milliliters, not a huge amount. And then they applied eDNA metabarcoding.
Okay, let's break down metabarcoding for everyone.
Right. So you take that water sample, which has this whole soup of DNA from everything upstream. You extract all the DNA. Then you use high-throughput sequencing, but you target just one specific short piece of a gene that acts like a barcode.
A genetic barcode.
Exactly. In this case, they used a standard one, a fragment of the COI gene, cytochrome C oxidase I. It varies enough between species that you can usually tell them apart.
Oh.
You sequence millions of these little COI barcodes from the sample, and then you compare them against huge reference databases to see which organisms they match, hopefully down to the family or even species level.
But how did they know the results weren't just noise or
Yeah.
contamination? You need a comparison, right? A control.
Absolutely essential. So at the very same 8 sites, they also did traditional kicknet sampling, focusing just on the aquatic macroinvertebrates.
Okay. The bugs.
The bugs, yeah. That was their gold standard for localized richness. It gave them a direct head-to-head comparison, the established local method versus this new integrated eDNA approach for the same group of organisms.
And what about the lab process itself? Tiny amounts of DNA sounds tricky. Did they check their own work?
Oh yeah, they were rigorous. They used what's called a mock community. This is basically a cocktail they mixed themselves with DNA from 33 invertebrate species they knew were in there.
Ah, known mixture.
Precisely. They ran this known mix through their entire workflow, the DNA extraction, the sequencing, the computer analysis, the whole pipeline.
And the result?
They found that 99.97% of the DNA sequences they got back were correctly assigned to the species they knew they'd put in.
Wow, okay. That's incredibly accurate. So the lab methods and the bioinformatics were solid.
Yeah, that gave them and us a lot of confidence in the actual river results.
All right, so the methodology checks out. Let's get to the big reveal. What did those water samples from the Glatt River actually tell them? What did they find?
Well, the sheer amount of life detected was, frankly, stunning. From just those 8 water samples, they identified 296 different families of organisms.
296 families?
Yep. Spanning 19 different phylostomes eras. Those are major branches on the tree of life. I mean, that's almost 300 major groups found in, essentially, a few bottles of water. Most of them, 196 families, were arthropods, insects, crustaceans, spiders, that sort of thing.
But the real mind bender, the thing that really screams conveyor belt, was finding things that don't even live in the water, right?
Exactly. That's the kicker. They didn't just find the usual aquatic suspects. When they looked at the species they could confidently identify and confirm were actually found in Switzerland.
Aha.
A huge number, 255 confirmed species, were terrestrial taxa, things that live on land.
Hold on. You're saying the river water told them about birds flying over or deer walking along the bank maybe miles upstream?
That's what the data showed.
Yeah.
It proves the river is acting as this collector, this integrator for the entire landscape surrounding it. DNA from land-based life, maybe fallen leaves, animal droppings washed in, carcasses near the bank, it all ends up in the river.
So the river isn't just a conveyor belt for its own residents, it's sampling the whole neighborhood.
It's a holistic landscape sampler. It's pretty revolutionary when you think about it.
Okay, let's do that direct comparison then. For the macroinvertebrates, the bugs they also sampled with kicknets. How did eDNA stack up against the traditional method for local richness, what scientists call alpha diversity?
eDNA came out significantly ahead in terms of sensitivity. At each site, on average, the eDNA found between 23 and 40 families of these invertebrates.
Okay.
The kicknets, at the same sites, only found between 17 and 24 families.
So eDNA found more families overall at each spot.
Yes, and interestingly, 32 families were ticked up only by the eDNA method. They were completely missed by the kicknets, whereas only 13 families were unique to the kicknet samples.
That's a pretty strong argument for eDNA sensitivity, but as you said, the real proof of the conveyor belt idea comes from the spatial pattern, right? The beta diversity, how community similarity changes over distance. This sounds a bit complex.
It is the crucial piece of evidence, yeah. Let's break it down. With the kicknet data, as expected, the further apart 2 sampling sites were, the more different their macroinvertebrate communities looked. Community dissimilarity increased with distance.
Right, that makes sense. Sample a spot here, sample another one 10 kilometers away, you'd expect different bugs. Local sampling gives you local results.
Exactly. That confirms the kicknet is giving you a localized picture. But here's the punchline. With the eDNA data,
Yeah.
community dissimilarity did not increase significantly with distance between the sites.
Whoa. So the mix of invertebrate DNA looked pretty much the same whether the samples were taken close together or far apart. How does that work?
Think of the river as that giant washing machine again, constantly tumbling and mixing. The downstream flow collects eDNA from all the upstream sources and blends it together.
Ah, Okay.
So that continuous transport and mixing essentially decouples, as the
Hmm.
authors put it, the detection of a species' DNA from its actual physical location right now. The DNA signal you pick up might have originated from an organism living, 200 meters upstream, or maybe even based on their system up to 12 kilometers upstream.
12 kilometers. That's just,
Right.
that's quite a distance. So the DNA signal is like an integrated history of what's been happening upstream.
Precisely. It's a catchment level signal.
But that immediately brings up a huge caveat, doesn't it? If the DNA could have come from 12 kilometers away, you can't point to your sample and say, this specific species lives right here.
You absolutely cannot. And that is the critical interpretation point. It's not a map of local presence in the same way a kicknet sample is. eDNA tells you a species is present somewhere in the upstream catchment contributing to that point. It's an integrated signal over potentially quite a broad area. Understanding that difference is fundamental.
Okay, so it's a different kind of information. But if we accept that, the potential for large-scale conservation seems massive.
It really is. It gives us a way, maybe for the first time, to get a reasonably accurate estimate of biodiversity richness for huge areas, I think, entire nature reserves or whole river basins. Because it naturally overcomes that chronic undersampling bias of point-based methods. It's a much more robust census tool for large scales.
What about more localized stuff, like, say, restoring a damaged section of river? Does this integrated signal help there?
Actually, yes, quite effectively. The paper discusses this. They estimated transport distances for eDNA could be up to 4 or 5 kilometers. And that distance happens to line up really well with the scale ecologists think is important for local species pools influencing recolonization.
Ah, I see.
So if you restore a stretch of river, eDNA taken just downstream can tell you which species are basically on deck upstream, ready to potentially move into your newly restored habitat. It helps set realistic goals.
That's clever. And thinking back to those terrestrial species they found, it feels like this could bridge some gaps between different fields of ecology.
Definitely. Historically, you often have aquatic ecologists doing their thing and terrestrial ecologists doing theirs, sometimes quite separately.
Right.
But if a single river water sample gives you data on the fish and the mayflies and the birds and mammals along the bank, well, that provides a unified data set for landscape-level ecology that just wasn't easily possible before. It could help bring those fields closer together.
It's a powerful tool, no doubt. But the authors were also clear about limitations. What are the main things we need to keep in mind? What doesn't it do perfectly yet?
Well, the first big one is the genetic marker itself. The used COI, which, as we said, is a good barcode for many things, especially invertebrates.
But not everything.
But not everything. They explicitly noted underrepresented groups like fish, flatworms, and diatoms, simply because COI isn't the ideal, most variable marker for those particular groups.
So the dream of one single test for everything isn't quite here yet. You might need multiple genetic markers, multiple tests run on that same water sample.
That seems to be the reality, yeah. One water sample in the field, but maybe multiple different genetic tests in the lab, depending on what groups you want to comprehensively cover.
Okay, what else?
The other major hurdle, and it's a big one, is the reference databases. You sequence millions of these DNA barcodes, but they're only useful if you can match them to a known species in a database like GenBank or BOLD. And those databases, while huge, still have gaps. They found quite a few sequences they couldn't confidently assign because there wasn't a good match.
I remember they made an interesting point comparing the error rates, though.
Yes, that was quite telling. Because of the database gaps, they had to set a threshold, and they ended up accepting what they estimated was about a 14% false absence rate, meaning they probably missed detecting 14% of the things that were actually there, but lacked a database match.
Okay, 14% sounds like a fair bit.
It does, but then they contrast it with traditional morphological ID using kicknets. Error rates there, misidentifications are reported to be anywhere from 22% up to nearly 34%.
Wow. So even with the database gaps, eDNA might still be more accurate overall than physically sorting and identifying tiny critters under a microscope.
It certainly puts the database limitation in perspective. It's a challenge, but maybe not as bad as the existing alternative in some cases.
So if the river's a conveyor belt, mixing signals from potentially kilometers away, the big question for researchers now is figuring out just how long that conveyor belt is in different situations, right?
Exactly. That's the crucial next step they highlight. We need more in situ studies experiments out in real rivers to measure the specific transport distances for community eDNA. How far does the whole mixed signal travel under different flow rates, different water chemistry, different temperatures, UV exposure.
Because it probably varies a lot between a fast mountain stream and a slow, muddy river.
You'd expect so, yeah. Nailing down those transport distances in different systems is key to confidently applying this integrated signal for specific conservation or management goals around the world.
It really feels like this paper forces us to see rivers differently, not just as pipes carrying water, but as these massive flowing information networks.
Yeah, dynamic databases capturing the life of an entire watershed.
It fundamentally shifts our thinking.
It really does.
Yeah.
And it challenges that whole concept of local presence in flowing waters. If your sample reflects a community stretching kilometers upstream, what does local even mean anymore for management?
Right.
How should we define conservation boundaries or set restoration targets? This science suggests we need to think not just about the site itself, but about that upstream species pool, potentially 5 kilometers away or more, that's constantly contributing to it via this conveyor belt.
A much bigger perspective.
And ultimately, unlocking the full power of this amazing tool comes back to those databases. We need that continued, really close collaboration between the field ecologists, the molecular folks in the lab, and crucially, the taxonomists, the experts who identify and catalog life to keep building and curating those digital reference libraries.
Digitizing life, essentially, turning physical specimens into reference sequences.
That's the key. That foundational work is what will let us truly leverage this incredible window into biodiversity that rivers provide.